weird. In like 2 weeks, I'm getting a rental car because my car's got to get fixed. Somebody hit and run my fender.
I know I'm tired. Since I took my bag out to the car after I showered. I left my damn bag in the car, my book bag. I'm not going to grab it right quick. Okay.
Whatever. Uninsured motorist, deductible. I'd be glad he kept going.
All right, look, guys. Hicks is going to go get his bag. So it's going to probably take, what, 5 minutes? So 5 minutes, we're going to get this bad boy started. We got a lot to talk about. Lots to talk about.
Like IGEs.
Yeah, I don't know. We're going to kind of go there today. I don't know that we're going to make it there today. I need to set it up before we get there.
Yeah. Solid plan.
Because before you get to the IgE, you got to kind of understand the inflammatory process and how antigens are made. And that's where we're going today.
Yeah.
And hopefully,
that antigen's got to be in place for that IgE antibody to come along.
absolutely, it's got to be there. Hey, Gwen and Maze, y'all doing okay this morning?
Yes, sir.
Good deal.
That's why I read the chapter first. No, so in 2 weeks I'm getting a rental car because my car's got to get fixed. And if the letter's still in the on the rental car, then that's going to be really weird. Because how much is he like watching me following? Like if that happens, then I might actually follow. Because it's.
Do you save them?
I mean, I just throw them in the middle of the dash. I put the car out of the weekend. I throw them away.
I don't throw them away because when you're dead, that's evidence.
You're laughing, but that's what my wife wanted the pictures. It's like if you come up this and going, I'm gonna show that picture to the police first. Dude, it's creepy, man.
You read the chapter?
I read up till selling.
You heard anything about extravasation?
What?
Extravasation?
Like a this, I know this.
Extravasation.
No, it's look at the back of the chapter.
I definitely didn't do that.
Nah, man.
You can't go there. Why ask you?
Dude, I was so confused. I got so hung up on those antibodies and started Googling IgE antibodies. And the only thing you can find,
I know what
I'm sure it's there somewhere.
To be extra.
Bradley, you say you never want to
It's in my way. I said, give me my number. He goes, give me my
come to the gym.
Yeah, why don't you start coming in here?
all right, punch ball. Something happened during this break, somebody go to bed. Talk.
Yeah, he's gonna kill me.
Maybe he's just lonely, man. He's just a lonely guy. He just wants to be your friend. Shit. Just wants to be your friend. Mark was gonna get a fire extinguisher to be his house. He's gonna show up. Hey, don't always need to be a part of.
Yeah, Bird's sick and left me there alone today. Yeah, they really. More actually called out.
So what is the start?
They're always like some float of some sort. No, it's not fun.
Why you gotta have tunnel vision in the gym?
Every day when I leave the gym and I'm walking in my car, I'm nervous. Because I know it's there. Well, I won't be there tomorrow. You're gonna find me a Monday.
Yeah, you know what?
I'll show my wife the pictures. It's like, oh, sweet Jesus.
Yeah, look at his teeth, man. He looks like a predator.
Dude, when we first started, you got
He's still that proud.
You cut your face off.
You would have came in and said, "Oh, it's skin on your face."
Found it. You're not making this better.
Found it.
Found it.
You need to give a crap.
Found it?
Black 14.
What page?
Extravasation?
Yeah. I seen it last night. I had no idea. But I couldn't.
Yeah. There's a reason why I'm upstairs on the treadmill.
I got to number.
I've never talked to him.
19. I'm 34 now.
Okay, Hicks back. There he is. All right. Let's get going. All right. What I want to talk to you today about, well, number one, everybody see the study guide I posted. It's on. If you look under the link for chapter 8, the study guide is there. All right. And so this might go a long ways in helping with your study guide today. We're at the part of the book where we're going to start talking about what happens when a cell's injured. What happens when a cell dies? And so we've kind of led up to it because, we know that an acidotic state can lead to cellular death or injury. We know that an alkalotic state can do the same thing. Cell death, it can lead to necrosis, right? So cell death can lead to necrosis. And if you look at your book, it basically states that necrosis can be caused by the cell being hypoxic for too long, right? It can, an injury to the cell can actually change the way the cell is shaped. And that's important because if the cell is like a different shape, you got to think about it now. Remember, cells make tissue, okay? See, these cells have all kinds of receptors on them and things they receive. That's how cells communicate with other cells. And so they have receptors on there. They receive enzymes of all different spectrums. So if that cell is a different shape, that might mean that a certain enzyme that cell needs it can't receive. And so we can say that we can look at cells and say the cell itself or the organelles inside the cell can swell. And there's something in your book, it's called lysosome enzymes. And so a lysosome is an organelle inside the cell, right? And so what the lysosome function is that when it comes up on when a white blood cell, which is the infection fighting cells, use this as an example. When it this is so hard to go over because the book has an abbreviated format here, right? And I don't want to go too deep. I just want you to get what you need. So if you see me up here struggling, it's because I'm struggling to try to present this in a way that meets what the book provides and we all understand it. But a macrophage, right? A macrophage is a certain type of white blood cell. And what's so cool really about that macrophage is that if it comes up on something that's foreign, right? If it comes up on a bacterial cell or a viral type cell sometimes, it can actually engulf that, which means it can actually eat that cell and bring it inside its cell. And once it's inside its cell, the lysosomes, and I like to think of it as lysosome. Yeah, so I remember, because that's what it does. It's inside now, the lysosomes will attack that bacterial cell that's inside and destroy the bacterial cell wall, releasing the contents of that inside of the cell. So that's what a lysosome does. But when you have cell death, those lysosomes can actually be ejected out from that cell and act as a mediator sometimes. And so when we talk about mediators, these are things that are produced by hypoxic cell that can actually damage or even possibly kill other cells. So we'll talk about some other mediators today. The book inside that section of cell death talks about the number one, because it's the number one mediator. And does anybody know what that is?
Free radical.
A free radical, right? There's free radicals that we know about actually probably pretty well. Does anybody know of what a free radical is that we use? No, well, just anywhere. You probably have some. You may have some at your house.
Virus.
No, it's not a virus. It's something that we actually use. Your parents may, if you're really old school. No, this is something that's not. It's not a bacteria that you have that we use. It's something that you can use if you have a cut or something. Hydrogen peroxide. Hydrogen peroxide is an example of a free radical, right? Because its chemical structure is H2O2. And so to make it really, easy, or not easy, or maybe a little bit easier to understand anyway, and what it means is if we have an atom here, right? And so let's say it's supposed to have 2 electrons. All right. Say it's supposed to have 2 electrons. And this is its outer shell. Right. And it only has 1 electron. It's missing 1. And so what this guy's going to do is it's going to come up, to the next guy who, may have the exact number it's supposed to have. Maybe this one's supposed to have, 4 electrons. And what it's going to do is going to say, hey, I need it. Let me have it. And it's like, but it's going to hurt me if I give it to you. I don't care. I want it. I want one of your electrons and I want it right now. And so when this happens, this electron goes away, but the cell becomes damaged because of a free radical. All right, so that's that free radical theory. And so just as a sidebar, where they really explore this at is for stroke care, because the brain happens to produce quite a few of these free radicals. And when somebody has a stroke, free radicals actually participate, are one of the villains in the process of destroying brain tissue secondary from hypoxia, right? Because in a stroke, what do you have? You have a you have a clot, right? That's blocking a cerebral artery, and that oxygen-rich blood can no longer make it to the brain tissue. Therefore, that tissue becomes hypoxic, necrotic, and then these free radicals start producing. And so here we have maybe a neurologist now who's going to try to make this better for somebody. So they're going to come in and try to go up into the brain, remove this clot. Well, sometimes they can do that, and restore blood flow, but those free radicals are still working at destroying brain tissue. And so that is kind of where this free radical, how it can be a problem. Yeah.
Are you saying those free radicals get past the clot and they're still?
No, they're before the clot, and because of the chemical reactions that are happening without that blood. And even when you restore the blood, the free radical's still there.
But they're the result of the necrosis.
Yes.
They traveled with the clot and they're, I see.
Yep, that is correct. So that's some stuff that, so even after you restore blood flow, a lot of times you're still left with having to deal with that kind of stuff. And so when you have this necrotic hypoxic state, that's just one of the mediators. Of course, then we can look at other things that damage cells. And that could be like a chemical injury. Yeah, so you could have for instance, pesticides can sometimes, they're nasty. I don't know if anybody's ever, sprayed the garage with bug spray because bugs are coming in the garage or something. So that stuff, you get too much, or you start inhaling too much of that stuff, and it's a bad day. I mean, you could start. I was doing it here a while back. Start getting a little bit nauseous, headache. I was pissed though. So I had some bugs and stuff coming in my garage. I called my pest control folks. They were coming out, but I was still upset. So I decided, I had some spray. I had to spray in the garage. That wasn't the best idea. So you can inhale some of that stuff and cause problems. But there's other things too that we really see quite a bit of in the wintertime.
Carbon monoxide.
Carbon monoxide, that's a chemical injury because of the affinity that hemoglobin has. It loves carbon monoxide. Loves it so much more than oxygen. I mean, loves it. Will kick oxygen to the curb. It loves carbon monoxide. Yep, smack gone. And so then instead of having the oxygen seated in the red blood cell, now we have carbon monoxide, and we can't diffuse carbon monoxide, right? I mean, I guess we could, but it's not going to work because we need oxygen. So there's an example of a chemical type injury. One of the products of combustion is cyanide. So cyanide poisoning is a problem for our for our career sec. You know,
Combustion of anything.
what's that? Combustion of anything. And so we are looking here at the cab, and I think we have got them now. Not quite sure. I think they're here. Cyanide kits. So in case you're in a situation where you are in contact with cyanide, we have an antidote kit for that, right? So because I mean it's happened in a few places around the nation where firefighters have died from that.
How many times can you use that kit?
Just once.
In your life?
Oh no, I don't know how many times you could use it. Like, I don't know. But there, you know, it's an example of a chemical type injury. So that's one. And then one today that we're going to talk about is an infectious injury. Okay, I'm gonna try my best to go into detail on that. Because infectious injuries can certainly damage a cell. And it damages a cell. And when it does damage a cell, it starts something called the inflammatory process to begin. And so that's kind of what I want to go over because it's confusing, right? It's kind of hard to get. It's hard for me to teach, but we're going to go there. All right, so let's start here. With a bacterial type cell. Okay, start here with this a bacterial type cell. All right, and so when we're talking about infectious type injuries, there's two things that we kind of look at. Right, there's the bacterial type of cell that can cause problems, and what's another infectious type of cell that's not a bacteria? Virus. Right. And so, and they're both very different in how they work. And they're both really fascinating to me. A bacterial cell is more of an independent type of cell, right? It can maintain its own deal. It has some organelles. It has, you know, so it's self-sufficient. Okay, it's not dependent upon anything other than the environmental conditions that it needs to thrive. Okay, so but then let's flip to the viral cell. Well, the viral cell is completely dependent upon the host. Okay, it doesn't have organelles. It doesn't have, as a matter of fact, it's almost compared as a parasite. Okay, it's an invader, but a very intelligent invader nonetheless. And what I think is so interesting about a virus cell is that it comes in and basically hijacks the cell at the nucleus, hijacks it, takes it over. And you know, the cell has the nucleus in it, right? And it has something called RNA and DNA. So DNA is what makes your little world go round, and RNA is just there to copy verbatim exactly the same DNA that's there. Because when you reproduce a cell, you need a carbon copy of that one cell. So RNA, the responsibility is like to copy it. It's the copy machine. All right, but anyway, it can rewrite the DNA. It can rewrite the DNA, right? And so one of the most famous viruses, in my opinion, the most famous virus It's AIDS, the AIDS virus. Because that's exactly what it does. It's if you had to, if somebody said you were an evil scientist and you were going to develop something that would kill millions of people, the AIDS virus is what I would have designed. Because it goes in. When you get it, you don't know you got it, number one. When you catch it, you don't know you have it at first. Then what it does is invade your cells at the nucleus level, and it decides to, it rewrites your DNA. It rewrites your DNA to the point to where it reprograms your T cells. These are your, this is the varsity football team.
They back up the leukocytes, or like the leukocytes first come in, right? They're the warriors, and then the T cells come in.
Yeah, so what happens is like your neutrophils and your macrophages come in, they're like the first responder cells. Your T cells are the ones who finish everything off. That's why. All right, so your T cells are your major infection fighting cells, right? And so it rewrites that DNA to where it says, hey, you don't see me, right? You don't see me. I'm here. You're not going to attack me. And then once it does that, it says, now I'm going to kill all the T cells. I'm going to kill every one of them. So it starts murdering your own infection fighting cells. And so now you've had this, let's say for about 8 or 9 months. Right? And so not only can you spread it to other people because you don't know you have it, now you get a sinus infection. Sinus infection. Nothing. But you don't have any T cells to fight it. Right? And now you have pneumonia. And you can't breathe. And you go to the hospital and they start pumping in antibiotics. But nothing's working because this is not a bacterial infection. It's a viral infection. And the bad thing about viral infections is just, like I said, it programs it to where you can't see me. So we don't have an antibiotic in the world that can kill a viral cell. And we've come a long way since AIDS virus. We have now antivirals, right? And we were able to actually use some of those antivirals in the corona epidemic that we've been going through. And so, like remdesivir is one. It's an antiviral. It's a really good one. But before, when AIDS came out, we didn't know what an antiviral was in 1980. And so people were dying left and right. And so that right there is an example of how serious a virus can be. So a virus, if you're looking at the anatomy of kind of like the anatomy of a virus, you look at it, it has, like I said, it has that RNA, it has DNA, and the outside structure of that virus is called a capsid. Okay, a capsid. And what it does is it protects, it protects that virus. It's a protective capsule, kind of, and it protects from antibiotics. I already talked about how it protects from antibiotics, but yeah, it's just a protective type capsule that protects the actual virus itself. So a virus, then if you're looking at viruses, so does it attack the extracellular portion of the cell, or is it the intracellular? It's intracellular because that's where your nucleus and all that stuff is. All right. All right. So let's talk about what we have here today. All right, so here we have this bacterial type cell. All right, it's over here. we're gonna say these are skin cells. And this bacteria, we have, well, however we got it. Could have come in through a cut. It could have come in through whatever. And so this bacteria is going to start attacking some of these skin cells over here, right? So it's going to start attacking these guys. And in response to this attack by this bacterial cell, it's going to make something. All right, and so what it's going to make is arachidonic acid. And it's in the book, this arachidonic acid is. That's the reason why most of the stuff, if I'm writing it out, I found it in the book. All right, and so now we're going to draw a picture of something here, of a different guy. Of a different cell. And this cell is called a mast cell. Mast cells are just out there on patrol, like a uniform officer in a patrol car, and they're just out there patrolling, and they're looking for the bad guys. Am I still what?
Virus, you're still on virus.
No, I'm talking about this bacterial cell right here. All right, and so it's going to these skin cells in response to being damaged makes this arachidonic acid, and it starts picking up on this, right? It starts sensing that, hey, we got some work to do, right? So it starts picking up on that. And so when that happens, remember what I said, chemical mediators? And we gave the example of the free radical, right? But there's other chemical mediators that can be created, right? And so this guy right here is going to start throwing out some pretty rough mediators, right? And so let's mention what they are. Yep.
Is that arachidonic acid bad guy that's created from that?
It's not the bad guy right here. It's what this is well, it will, yeah, it does actually, yeah. It's the arachidonic acid being present, picked up by the mast cell, that's going to lead to all of this stuff. Okay. And so the first thing that we're going to talk about is histamines. Go ahead, Perez. There is, I'm sure there is, because it's going to, arachidonic acid just didn't show up. It was from a different type of something, so I can't think of it now, but yeah.
Yeah.
What's that? Oh, you're trying to find it in the book? Well, look at 342. Yep. All right, so we have histamine. All right, leukotrienes. Yep. Prostaglandins. And it's going to produce it's called kinin, but really it's your coagulation clotting system or your kinin system that gets activated here. So I'm just going to put that up there. It's just kinin, but you'll see it in your book as a coagulation clotting system, kinin system. All right, so that's what this is going to create. All right, and so here we are in this process. So how is all this going to affect us? Well, up here is a blood vessel. Okay, this is part of the vascular system, and just like any other type of cell, it's got receptors on the outside of the membrane here. We learned about those protein receptors, and all of this is going to start coming up here and reacting. It's going to start reacting and so what happens is it's going to make it inside the cell wall. It's going to actually make it inside the cell. All of this stuff is. And your body's pretty smart in how it's going to handle this. Because it's going to start putting up I know what I call them, but that's not the right name. It's kind of like a receptor. It's like a protein receptor, but I like to call them like flags. Like a I need help flag. You know what I mean? Like if you didn't have a phone to call 911, but what if you had a flag you could hang on your front porch if you needed help? Right? That would be what these guys do.
So once all of those things start penetrating that cell wall, then these flags.
This flag comes up, right? All right. So I'm, so that's where we're at right now. Not too hard to get yet. Not too hard to get yet.
What is the importance of the histamine, serotonin, and increasing the permeability come into play?
That's where I'm going right now.
That's how it gets in, right?
Yep. I'm fixing to go there right now. But I need some room to do it. So I'm just redrawing this up here so I can have some room. All right, so what happens here? So what is going to happen? So if we have histamines, leukotrienes, prostaglandins, kinin system, they're all in play right now. Remember, we got our flags that are up here. All right. So the first thing that's going to happen is these cells have contracted. They contract. All right, and you've noticed that they've contracted because of these gaps here. These are gaps. So if this is the cell membrane, and we've got gaps in the cell membrane, right? What's going to happen to the fluid inside here? It's going to leak out, right? And it's not big enough to let the cellular fluid through, but it's big enough to let the plasma through, right? I'm going to get there. And so if that's the case, right, if we have fluid leaking through there, it's called increase what? We're increasing the perm. Yes. Right? We're going to increase the permeability. So we're going to have stuff, we're going to have fluid leaking out here. Well, okay, so we probably should really, before, we probably at this point in time should really define what inflammation is, right? So what is inflammation? Huh? Nope. Inflammation is tissue damage that's caused, it's tissue damage that causes a cellular response, right? Tissue damage that causes a cellular response. And so we had tissue damage down there at the skin tissue cells were damaged. So we have a cellular response going on. And let's talk about the signs and symptoms real quick of inflammation. One is swelling. The second one would be pain. A third one could be heat. And we could have redness. Thank you. Redness. All right, so swelling, pain, heat, redness. Well, if I have fluid leaking out, right, does that meet the definition of edema? Sure does, doesn't it? And so let me draw this out here real quick. It's okay, we're gonna make it. Where are smooth muscle cells located? Yep, inside blood vessels, right? Right, inside, they're also located in the bronchioles, right? So another response from these histamines, these leukotrienes, and prostaglandins is that it's going to cause these smooth muscles to relax. And and what's another word for that? Thank you. Dilation. All right, so that's what's happening here with this vessel. It's dilating. We have smooth muscle dilation. So the vessel itself is becoming larger. I'll explain why in just a second. And so the vessel's becoming larger, and our membrane is feeling the effects of this because it's contracted. So we've increased permeability, and we have a fluid loss. And so that pretty much describes edema to a T, right?
Fluid loss in this situation, you're saying you're losing fluid from the vessels, not from the cells, right?
Yes. Yep.
Cells are swelling out.
This is the membrane of the blood vessel. All right. So what this can be used for to understand better is an anaphylactic reaction. It's the same process, except it's a little different when we get there, but these same players are involved. It's the same thing. So it doesn't matter if I burn my arm or get a cut, or I have a bacteria here. The body's responding to that cellular injury in the same way. Make sense? All right. What's that? I couldn't hear you.
It doesn't.
Well, it's okay. Dude, yeah, well, I hadn't got there yet. I hadn't got to those flags yet. It's good. Just soak it in. It's like acid base. You gotta don't try to figure every little part of this out yet. First off, skim it. I just let it soak in, right? And then we'll go right back over this. This won't be the only time we go over this process. I'm sorry, I got Chief Burrell text me.
He's those YouTube videos you keep sharing.
Yeah, oh, the mustache guy?
Yeah, the mustache guy.
Yeah, that mustache is so nice. Did you see the guy downstairs getting ice yesterday? Did you see the stash? I think it was gorgeous.
All right, I'm sorry about that. All right, so yeah, just let's soak it in first, and I'm gonna go right back to it. I don't expect you to get this like immediately. It's stuff's hard. But let's just keep going with it. All right, so smooth muscles are going to relax because of the response to the chemical mediators. All right, that is causing vasodilation. I'll get there in a second. And so, but we've explained why you have swelling. Check. Now, let's talk about the pain part. So it's easy to figure this part out, I think. Man, black again. All right, so these guys right here are called pain receptors. When they're located, throughout your skin, that dermis level, where the nerves are and all that. Okay. If I start dumping a bunch of pressure on these guys from edema, right? Is it feasible to understand we'd have pain from that? So, I mean, all you got to do is think about this. All right. So you got think about that square diagram that's in your book that has epidermis, dermis, right? And the basement part of that as well. Think about your burns. First degree, it's at epidermis. Second degree is usually at the dermal level, right in the middle where all the nerves are at, right? Well, think about then, if that's where all those pain receptors are at, if I start pushing down on them because of edema, fluid, and it starts coming down and squeezing them, that's what their job is to do, is to start hurting. So they are going to respond to that with pain because of edema. So that's your pain. All right, so heat and redness. If, for instance, it's better probably to think about this like a cut on your hand or something like that, or or burn maybe. But if this vessel right here is dilated, right, and I'm sending in, I'm trying to fix it, and I'm sending in more blood volume, right? I'm circulating that blood quicker to try to fill the container. I'm increasing the amount of blood that's normally in that area. If that's the case, and I'm increasing more blood that's around in that area, what do we know about blood? Yeah, it's warm. It's warm. Yeah. It's warm. So that's going to increase. That's going to increase that heat in that area, and blood is going to cause that area to be red too. Right? There you go. There's your
This
heat.
is all Lucas's fault? Or
This is all these guys' fault. Leukotriene, histamine, prostaglandins,
it's all their fault?
all their fault. I know you want. They don't either. Don't fight them right now. They'll be okay. I know you want to go bow up on them, get locked in on them, but no, it's okay. It is their fault, but it's all right. All right, so let's then talk. Let's go back up here then, since we kind of explained that sort of. Let's look at why this vessel needs to. Why? Well, we know why it's dilating, but sometimes the body responds with vasodilation, and it's not a bad thing. Sometimes it's in response to what it has to do to try to fix this problem. Okay, and so what we're doing is we're making this area a little larger because we got some folks here. We got some folks here. Okay, we got to get some people in here. All right, so this is called a neutrophil. And this guy right here is called a macrophage. All right, neutrophil and a macrophage, and they're both white blood cells, so they're effective infection-fighting cells, right? They both can participate in something called phagocytosis, and phagocytosis means eat cell.
That was lucrophils and what else got in up there?
Yeah.
Sorry.
No, it's
Those
fine.
two names. Macrophage.
Yeah. Oh, these guys right here? Yeah.
Yeah.
Let me name them out here real quick. Neutrophil. And this guy right here is a macrophage. Yeah. That's what everybody compares it to, is a Pac-Man, but it looks like that.
It's the neutral symbol.
It's just, I don't know.
Yeah.
That's just it. All right. And so neutrophils and the macrophage, right? And so, you know what's cool about them? You know what's really cool about them?
What is this?
Tell me.
Does that look familiar?
Flags.
Oh, they do.
The war
And does that look like it will fit there?
Yeah, boy.
It will. It will.
Oh, shit.
It will. All right, so we'll just use this guy as an example. Both of them have this, and they are going to hook on right here. They're locking in. So what happens is this area is bigger so that we can get some neutrophils in here and some macrophages in here. Because they're getting their ass beat inside there. They need help. They can't fight this. They can't fight this. They have got to get some help in there. And that's what these guys do. They come by and that grabs them. It says, oh, we got a problem. Don't you go by us. Don't you dare float by us. We got issues. Right? We got issues. And so what happens is, but when it starts slowing these guys down, it changes the way they float through. It's going to change the way they float through. So instead of just floating in, once these guys get caught up, they roll through. They start making them roll different, right? And it's that rolling, all right, it's that rolling that we call margination. That's important. It's in your book. And if you read that definition, you probably wouldn't get that definition. It's probably hard to understand this from reading it. But that's what margination is. It changes the way these guys move through. All right, so all right, let's take 15 minutes. Come back. Go ahead.
Back up. Where did, how did and where did neutrophil and macrophage come from?
So they're in the body.
Right.
They're floating around just like this macrophage is.
Right.
It's just floating around as part of your white blood cells.
Okay.
Right.
Those two things are part of the right blood cell.
Yes.
So they're ready to show up and fight regardless.
Right.
Once all this happens, they're instantly triggered.
They are.
They're on the way.
They're on the way.
Okay.
And we're going to talk more about how they're triggered.
Right. Which was the same thing as white blood cells fight infection. They're part of,
They are white blood cells.
deeper portion of the white blood
Right. They have, if you take a white blood cell, we can break it down to different types of white blood cells, but they are white blood cells.
I just couldn't figure out where they came from. You threw them up there. I'm like, how'd they
Right.
show up?
How'd they show up?
Where'd they
Right.
get from?
So yeah, remember we're losing plasma, but we're not losing cellular blood. We're not losing white blood cells. We're not
Oh,
losing.
so they're still in there.
Yes,
They were
they're
already
still in
there.
there. They were
never
already
got
there.
pushed out.
They never got pushed out.
Also, they didn't come in.
Right, because because when we lose plasma, that's the water part of the blood. That's cellular blood, red blood cells, white blood cells, and platelets. They can't leave. When we don't lose all of that plasma, we're just losing some of it.
I got it. I'm kind of.
Seriously, class, I have no extra time outside of it.
So far, all of this that's happening is in response to the body kicking its own ass because of bacteria.
Absolutely. Now, it's a good ass kicking.
Right.
At this
Something's
point.
gonna happen.
When we talk, I said, anaphylaxis is the same process. It is, except it goes ham. Right, it goes ham. Instead of making the normal amount of prostaglandins instead of normal amount of leukotrienes and all that, it goes ham. It starts making 4 times more than it should.
I'm gonna go ham on Barker later.
How are you?
He just doesn't know when it's gonna happen.
Well, yeah, that's a good thing. Always attack your enemy by surprise. Barker's one of the type of guys, though, like he would go everything you had on him, and he would stand there and go, "What are you doing to me?"
Well, normally, yes,
And
but
we'll come back for more.
there is an off button in all.
He would be like, "Are you just playing or what?" And you're giving everything you got.
This is actually good.
So
Yeah, like, so what's the off?
that's a massive. Okay. And in your book,
Yeah, man, I gotta be on
it
Saturday.
tells
Well, I gotta have this.
Yeah,
before I leave.
yeah.
When I leave here, I got 9 minutes to register. So I'm in the fucking zone. Oh, I don't know. I don't get more. At that point, I wouldn't even ask. Yeah, it's $100 or no job. And I'll big money third.
Oh, yeah.
You gotta tell me, yeah, look at it.
Joe Biden said it.
Oh, it's true.
You know, Joe Biden said it.
Hey, and you, it's,
gotta get one more step.
Yeah,
What if they say you can't get the next 2 years?
Oh, yeah.
Get a virus.
I can.
I'll get it. I've done that for the first 20 miles. Yeah, you know, it's important. You gotta look out. It's all about herd and, I'll be the first. It's all about it's a group effort. Come on, let's do it. Well,
You've seen the show, right?
Yeah. So, set.
I bet you next week they'll be like, yeah. Oh, you did?
Yes, I did. Because he said.
Oh, Brad.
My second one.
Next week.
Like, I don't know why he said first.
I mean, I was going from my mom. Oh, they'll have to die. I'm getting that I'll meet the movie. He'll do that.
Yeah, he'll do that.
He'll do that right now.
There's something there. You won't go.
No red light. That shit was funny. Rather keep up the balance show for you.
Right. Hey, what's up? Bradley will fuck him like the guy did on Snowfall. Put me on him. I'll scare him away.
I don't.
He'll find out what he
you
wants.
Oh, yeah, baby. He'll leave the cab county for good.
But yeah. Who's is that? What are you comparing it to? Oh, I mean, you got soda.
He's a big.
Huh? They're themselves, what?
Chicken salad.
Chicken salad.
Hey, we just put out things that you thought the trade deadline got crazy yesterday. Doesn't end till 4 today.
No, it's, I think the break can get too fine.
Really? Dude, the Cubs are jumping, bro. They're jumping. They're starting over, dude.
That's why I'm saying.
It's funny, man.
That would be awesome.
Same in that color.
We get pictures like show the neuro shape.
I never got it.
Dude, I'd love to see Chris Bryant here. But I want Duval back too, though.
He's not coming.
I seen that last time.
Yeah. Turn it right over.
I guess that's
Yeah, but there's still some things in play, because the Marlins got a package. So, I mean, Duval released.
You're gonna end up signing. Maybe like how
Yeah.
it is.
So I'm saying they got a catfish.
If they do it right, they can sign Duval.
Well, what could happen today is the Marlins want somebody. And the first place they're gonna go is to Atlanta to dump the ball. For sure.
Always gets dumped in Atlanta, dumped the year after.
But that'd be their first call. That'd be their first way to clear up space. They want to, well, yeah, but they, well, I mean, take Brian. Yeah.
That would be a. I don't think he's doing it.
Oh, he's supposed to do deal after this.
The Braves have to save money because the Braves knows that. Contract, yeah,
We're okay. We're okay.
which is fine, which makes me have like usually like Yankees, some years steal, like big players like bro, they just got rid of the
Ravens come out and said he's staying with them.
all those.
Yeah.
Unless we don't go back.
All the Ravens at that, and he's taking he's a loyal. It's not even. Raven will say less. He's a loyal.
Because what he does, he knows that we already have policy the next 10 years.
Yeah.
He's not going to. If he went anywhere, I'm going to go back out to California. So he'd probably try to play the Trout or I don't know if he'd like the Dodgers. He might go to the Dodgers.
It's hard to say. The job fusion is not the better we don't need anywhere.
Bro, this is cool technology, man. It's looking like in all. It's astonishing.
Hey, by the way, I think I'm still not giving people. It's still so bad. Bro, did you see that one yesterday? 450. He would have had raised line. I'm gonna keep it. You want a tiny with 5 more seconds.
Because I want, and that's how I want those sports. Well, maybe not.
That's all, that's next.
Oh, yeah. I don't want to keep up. Well, I'll just give it about 3 more years.
The first run online, I was like, at first I was like, I don't know if I really like it. I was like, he was like, I I'm making no profit with like $16. No. So I went to one. You probably might tell something. Who's that picture? I'm selling to myself. When I was a crowd, I remember the answer was like You want to stop? Which one? Just the one that
The problem is that is normal. The problem is I gotta do it. It's like I said, I use like with my fingers as a
Hey, Campbell.
Yeah. But the thing is, man, come out here with me. I mean,
Think about how mass produced. Not a lot.
Hey, give me just a minute. We're going to figure out.
Yeah, how you doing?
still bad. Oh, no.
Were you there? Did you pick up? Not important. We're there from the beginning. Start with chapter 7. I feel like that was yeah, my problem is like I'm tired of there. Like I like a lot of people left, so I feel like there's stuff that I need to know more. This, yeah, he does. And that's the only thing about it right now. That's like, yeah, I guess because I get that. I start hearing more. I don't do like I watch it, but if I draw that picture, I'm getting it. So I write it out. Everything I'm saying. I write the process. But then he keeps saying yes, all the good stuff. You know what I mean? Instead of low and go back and explain. Like give me the I think a lot of people that is so easy to it's not like yeah, but the thing is, you know, he explains that so you're not gonna get it. You gotta that's what he's telling when I get the end of it, I'm like, you're probably going I'm that's, I mean, otherwise he just said infectious injury. I'm getting the honestly I had no no, because sometimes for some days you're not right, but I can go home. You know what I mean? Like I can be out of it, go home, and still be, you know what I mean? But I don't have that time. But I'm gonna tell you another thing. Even if you don't get this, if you go home and come back to the started that, you make more sense. We got no right now than people who didn't. Is this fluid leakage?
Yeah.
That's kind of what extravasation is.
What page you on?
328.
Extra elevation.
It's a huge chapter. Nine is just like feeds a bit. Quick and easy. They were cruising. Which is beautiful. That's my sure. And I can just stay on.
I went through it, so it's not.
2 weeks I can prove it hard. And this that's the property.
I saw it last night.
Hey, Gwen and Maze, y'all still there?
I'm here.
We're just waiting on Jones to come back in. And once he gets back, we're going to start back.
Okay.
As soon as you said it that day. It's that bonus.
So where? Oh, you wait for it? You going to on the lake or one of those places? Which one? Rome, Cartersville area? That big one that's got the complex where it's like you can see it over that hill where you drive up that major highway. All the ball fields and stuff. That one's sick. That one is sick, dude.
I don't know anyone that has $100,000 boat, so.
Yeah, there's that. That's cool. That's sick.
Back out too? Oh, sorry.
I can't stop being a bitch.
Either other way, those are perfectly those family or something that happened to your life. God,
It's not that far.
When you guys get first, what? When you guys get first, like 15 minutes.
Who else going? Who else going?
It's the girl that broke the other.
And it's funny, looking at I feel like I was killing myself last night on the review. And now if I just waited till today,
Yeah, I know, right?
there's so many more I'm gonna be able to fill out tonight easily.
Bring some water and some sunlight.
Okay. All right. Y'all ready? All right, so we left off right here where we're talking about bringing these white blood cells to the fight, right? And we just explained that when these white blood cells are coming through here, that the way they come through the vessel with them rolling through is called migration. All right, and so now we need to bring these white blood cells to the fight. And that's what we got here with our flags, right? But we need them down here where our bacteria is hanging out too, right? And so what's kind of cool about this is these white blood cells, when they get into down here into this fight, they're fighting now. They're going to come out from this intracellular space, and they're going to be pulled down and squeezed through these gaps, right? These gaps where the cells have contracted, right? Remember that membrane is more permeable, and it's going to allow these white blood cells to come out. And this has a definition. All right, so this has a definition, and it's called transmigration or diapedesis. Okay, diapedesis or transmigration. That's when the white blood cells, the macrophage, and the neutrophil are going to be pulled down through the gaps right through those gaps. Sure, that's fine. When the white blood cells they're flagged up here, right? They're rolling through a process called margination. But now they're in the fight, and these white blood cells, they need to get out of this intracellular space, out here to this extracellular space where the bacteria is. And so they're going to get pulled down through the gaps where the cells have contracted, where the membrane is more permeable, through a process known as diapedesis or transmigration. Mm-hmm.
Those two things inside that box again, I can't read that.
Yeah, neutrophil and macrophage. Hang on, I'll try to make it bigger for you. All right. So here we go. Let me take over here. Let me make us a little bit of room so we can talk about this next part. I heard you, Sue. Give me just a second. All right, go ahead.
So the point of those flags then is just to get the white blood cells to start rolling to get them into the roll time so they can roll out of the vessel.
Right, it's to get them, bring them into the fight. So there's another definition in your book that we need to cover. Okay, so what we have now, right? So remember, this bacterial cell is putting out those mediators. Okay, so chemical mediators are coming out of this cell, right? And so it's going all over the place out here. But what it's going to do is it's those chemical mediators that the white blood cell is going to be attracted to. So these guys make it out of, around these, through the permeable membrane, through the gaps, right? Diapedesis. They're out here now and they're looking for this bacteria. They got to find it. And the way they find it is those chemical mediators that this is putting off, it grabs their attention. So instead of these guys, going the wrong way and floating through the fluid, that way, it's going to stop them. It's like somebody threw a baseball or somebody did something to hit them in the shoulder. It grabs their attention. It stops them and changes their direction to go move towards the bacteria. Okay, that is called chemotaxis. You look on page 343, you'll see chemotaxis.
Almost like those mediators with just little magnets for those things.
And so during this process, let's say these white blood cells, they get over there, right? They get to the scene of the crime. And just like a police officer, when he rolls up on somebody and he anticipates a problem, what does that police officer do? If he's by himself, he calls for another unit. So these white blood cells are going to do the same thing, right? They're going to call for more white blood cells, but they're going to release a chemical to do that, right? And that's called interleukin 1. Interleukin 1. So that will attract other white blood cells to come to the scene and help with this process.
And that just happens instantly.
Yep, as soon as these guys are in the fight and they make contact, they start releasing interleukin 1.
Make contact, they calls.
Yep.
More of the same type of cells.
Yes, more neutrophils and more macrophages.
What is it that bacteria cells are putting off the white blood cells?
Chemical mediators. Yep. So there's one more thing I'm going to talk about, and then I'm going to back up and go right back through it again. Okay. And there's more to talk about after this, but for today, this is stuff I want you to get. All right. So interleukin 1's been released, and that's going to call those other white blood cells. All right, so we're going to look at like this macrophage for a second, talk about how it does its thing. All right, that's kind of jacked up a little bit, but I get my point here. All right, so here's your macrophage. And your macrophage and your neutrophil can both operate under something known as phagocytosis. So what it's going to do is going to come up on that bacterial cell and it has these little tentacle looking things like that. It's just really changing the shape of the cell. It's going to latch onto that bacteria. And what it's going to do, it's going to pull it inside the cell. So now it pulls it inside. We have that. Now we have our, let's put our bacterial in there.
What did you say it operates on?
So it pulls that in there. And then we also have our lysosome in here. And this lysosome has all these little enzymes in here, inside that lysosome, that's going to attack the bacteria and rupture the cell wall of the bacteria to the point to where you have, it look like this. So now the cell wall of that bacteria is gone. So we'll learn about this later. But what this thing to think of is this, is that macrophages nor neutrophils are what they call antigen presenting cells. See, these now are antigens. And so what we want at the end of the day to get immunity from this thing is we want an antigen presenting cell, right? So you would have like a B cell or a, I'm sorry, yeah, that's right, a B cell or a dendritic cell. You know what? I think I said wrong. Macrophages actually are antigen. I was thinking of a neutrophil I was doing here, but I'm doing a macrophage. But anyway, we'll get to that later. But what at the end of the day, what you want is you want an antigen presenting cell that can take this, what's been broke down, and display it up here on the cell wall as a protein. Because at the end of the day, what we want is the next time this antigen shows up in our system, it's defeated readily. Boom, done. Go ahead.
So the macrophage doesn't itself eat the bacteria.
No, it doesn't. It breaks that cell wall down. So how we get, that's right. So what needs to happen is we talked about it a little bit. It won't blow your mind too much today.
what I was asking was, so you have a little macrophage up there up the top.
Yep.
and those small ones actually grabbing onto the
Yeah,
bacteria.
gotcha.
And then to the one on the right now, what's the blue line?
That is a-
That's regular cells, macrophage bring it into a cell?
Yes, the macrophage grabs this bacterial cell right here, pulls it into itself. Now this is a cell wall. Here's your macrophage. It's pulling this bacterial cell in there with it. And this is our lysosomes. And what the lysosome is going to do is break down the cell wall of the bacteria. So now this bacteria becomes antigens.
Right, so that macrophage brings itself and that bacteria
Yeah.
into the cell, and then
Right.
Macrophage is originally in the cell.
Well, no, the macrophage is a cell.
That blue part is part of the macrophage.
Yes,
Cell wall
this is part of the macrophage. This whole thing is together.
Oh,
all right.
oh, I see. So
All that's together is one cell. All this together is one cell. The only thing that's different here is the bacteria. The lysosome is an organelle, right? It's an organelle inside the macrophage.
Is that a little pigment thing an actual organelle, or is that just your representation?
It's just a representation.
So there's no.
It's changing the shape. Like, hang on.
Record that, right?
Yep. All right. Hang on. Let me show you all again.
Be clear, the macrophage. the type of
I'm
cell-
going to try to be clear. That's why I'm erasing this. All right.
Specific.
So if we have let me just redraw the macrophage so that it doesn't confuse anybody. All right, so all right, you see the macrophage is going to change its shape to where it can come in contact with this bacterial cell. And it's going to eat it. Because what we're talking about here is phagocytosis, eat cell. So then you're going to have your macrophage. Hang on, let me draw a cell wall so you'll know that that's still part of it.
that's what was going to be the part. Earlier, it seemed like the macrophage was inside something else.
So the macrophage now is getting this bacteria cell and going to bring it in. So now let's draw. Here's going to be our macrophage. This will be our lysosome. And let's start bacteria in there. Let's pick a little red for the antigens. Hang on, let me finish my thought. All right. And so now, let's put the cell, here's like our cell wall, sort of, kind of. All right, so now, you know what? That seems to confuse everybody. Let's just pretend like it's not there. All right. And so there is everybody in one cell. Now what's going to happen is this lysosome, love it when that happens. This lysosome, right, with all of its proteins in here, is going to bust down that bacteria to destroy the bacteria cell wall. So at the end of the day, what you're going to end up having is just this. You're going to have your lysosome here. All right, your lysosome, your antigens here. All right, and your lysosome still hanging out, I bet, somewhere up there. All right, and so now the cell wall is gone. No more cell wall. All we have is antigens. Now, what will happen is if this is not an antigen presenting cell, that means it can't put the antigen as a receptor. So if we're going to take something, we know that phagocytosis means bring it in. What's another definition for pushing it out? Let's do some exocytosis, right? And through exocytosis, we will send this out into the extracellular fluid. Well, isn't that dangerous? It's like releasing the criminal, right? Well, you know, the immune system is really awesome. But so now what it's going to do, maybe this will help you have a better understanding of this system, is the lymphatic system. That's your lymph nodes. Remember, yeah, it takes in fluid particles, right? And returns the fluid back to the circulatory system. That's what the lymphatic system does. So what it'll do is it'll grab these guys, hold them until an antigen presenting cell comes by to collect. That cell will collect, it will come by, and then it will put that bacteria that's now an antigen up here as a antigen. So if this ever comes back, we'll have a means to deal with it. So when we talk about the Ige antigen, what happens there is all of this happens, right? Except this is how we would take care of a bacteria, right? So this would be, that's the process. Well, if I wanted to take this into the world of anaphylaxis, right? I'm going to have that mast cell. We have an antigen. We've been stung by a bee, and we went through this process. Got stung by a bee, by a yellow jacket. We went through this process. The poison came in. Blah blah blah. The cells are like, hey, let's do something. We're protect ourselves against this thing. So it makes an antigen. Well, instead of now doing relatively nothing on the second time you get stung by a bee, right? Should be nothing. Been stung once before. It sees that this guy is here, and it's like, you know, last time you showed up, I didn't get to do anything, so I'm whipping your ass now. You're going to pay. And so remember histamine and leukotrienes and prostaglandin and the Keenan system. It is going to go ham. Instead of releasing a normal amount of histamine and leukotrienes, no, sir. It is going to go straight up. And so remember the cell permeability problem that we had? That's really not too big of a problem when you cut yourself and you have a little bit of an infection. It's not usually too big of a problem when you have strep throat, right? Not too big of a problem, but people have died from strep throat before antibiotics. Thousands, right? So, but not everybody died from strep throat, but thousands of people did. So it depends on how bad the bacteria, how your body responds to that during the inflammatory process. But with something like this, instead of a normal response, it goes ham. And that's why the body responds so violently to it with the upper, with all the edema and all that. Next lecture, we're going to go a little bit further. Next lecture, we'll talk about how the histamines and leukotrienes, prostaglandins affect the lungs, and we'll talk about how some other things that come about from this. But this is probably 80%. Go ahead, sir.
What was the content that was picked out from the cell?
Okay, that content is called antigens. Once phagocytosis comes in and pulls that cell inside, it's no longer a bacteria anymore, because those lysosomes are going to change it and make it an antigen.
Lysol cleaned it off.
Lysol cleaned it off, man. It turned it into from something bad to something good. So that you would have something called innate immunity, a piece of innate immunity. So that if you catch this again, you won't react to it as bad, right? So you won't respond to it so bad. It'll say, okay, I've seen this guy before. We dealt with him. We have an antigen developed. We're good, right? That's kind of how that works. So let's start from square one again. Let's go through it another time. Let's go through it one more time. Because this is really what leads into the anaphylaxis. Any type of severe inflammatory response. This is the stuff that you want to know about. And so this over here is like a skin cell. But look, I mean, cells are specialized, right? I mean, it could be any type of cell that's damaged that would cause this response, because cells make tissues. So we're talking about tissue, right? That's what we're talking about. So yeah, pretty cool. All right, so let's draw out our bacterial cell. Let's start attacking. So we have a bacterial cell. It could be whatever you want it to be. It could be whatever type. All right, it starts attacking this skin cell. Now, the skin cell what is it gonna make from that process? Yeah. Sure is. It sure is. All right, so.
Question.
Yep, go ahead.
Where is this bacteria in the body right now? Is it just floating?
It's just floating. It entered the skin. We could, yeah, we could say maybe you had a cut on your hand or arm and it bacteria landed and moon stars lined up perfect for it to sink in.
Same with the mast cell.
The mast cell is floating around as part of as a immune response.
That acid is not a mediator for bacteria.
Yeah, right. Well, it could actually be a mediator because it's produced by the skin cells. It can be produced by any type of cell that's damaged. So arachidonic acid can be a chemical mediator. All right, so there's our arachidonic acid. That's the first thing. And so we got a mast cell that's on patrol.
Does everybody have a certain number of mast cells? Or does that even matter?
That's the police right there.
Yeah, I don't know if there's a certain number or not of how that works. So the mast cell comes by, picks up on this arachidonic acid, and now starts making those chemical mediators. All right, and so we know that we're going to have some histamine, some leukotrienes, some prostaglandin, and all of these are in your book, so that's why if they wouldn't, I would have left it out. Prostaglandins and kinin. All right, so there's our histamines. Mast cell produces in response to this acid. And now these guys here are going to react. With the receptors on the outside of the cells. Now, like I said, there's receptors for these guys because this is not always a bad thing. Even though we're having some issues here, this is a part of the immune system. That's why we're sick. That's why we get sick, right? Is because of this. So there's symptoms to go along with this. But so here we are coming up here to react with the receptors.
And is it one of those or like basically all of those?
All of these are receptors, and there could be hundreds of them, thousands.
And the chemical mediators, like are all of those together, just looking onto one set receptor?
There's probably different, like histamine is pretty potent. Yeah. And so you might have more histamine up here than you have leukotrienes, but they're all basically attacking.
And each of them are like, are they all like one thing, or is it like?
Yeah.
Histamines connected to one.
Right. No, they're all one. They're all one. They're all one thing. So it's created, four things here, four separate things, and they're all their own entity and how they'll respond.
And they're all going through receptors.
Yep.
Written in black over histamine. What does that say?
Chemical mediator. I just tried to abbreviate it. All right, so let's go back and put our what we need to have if we're going to have, if we have an inflammatory response. So we know swelling, redness, what else? Heat. Pain. All right, so let's talk about then how these all come about. So the first thing that happens with this process is these cells contract. So if they're contracting, that means they're coming in close. Yeah, let's see here. So they're going to contract. And that's going to leave some gaps, isn't it? Leave us some gaps. All right, so you guys kind of get the idea here, I think. Cells contract, they become kind of gappy, okay? So this is going to do what to the membrane? So it's going to increase permeability. And it's that increased permeability that allows for plasma, right, to leak. All right, so plasma leaks out. And so we know too that the type of muscle that we have here is smooth muscle, right? And what does this smooth muscle, when it relaxes, what does it do? Dilates. All right, so we're going to have vasodilation. So there we go. And so now we can see that fluid, right? Otherwise, I mean, we could call that edema, right? So there's swelling answered. And so then we said, well, there needs to be some redness. So we said that since this vessel is smooth muscle, it has vasodilation, what does that do to the blood? What's it? I mean, besides, we understand we're going to have plasma leaking, but is that going to increase the amount of blood that comes through that vessel? Right. So we're going to increase the volume of blood. Blood is warm right? And so it's also that blood that causes redness, right? Because we've got a lot of it over there in that area. So that gives us the redness, that gives us the heat. And then we said edema pressing down on those pain receptors causes that pain. All right, so we answered that. We've got that going on. Now let's talk a little bit about, let's talk a little bit about what we talked about earlier. How that vessel responds when the white blood cells show up. So we got to put our flags up.
Sorry, this cell represents my blood vessels, my cells and blood vessels.
Yes, because I was talking about plasma leaking. That's the only place you're going to find plasma. Okay. All right. So here we have, we're still gappy, right? We know what's going on. We understand the edema, pain, warm, and redness. Let's talk about now how is this body going to respond to the bacterial part of this. There's our macrophage. There's our neutrophil. They're white blood cells, right? This is what they do. And now we got the flag here that's going to fit perfectly with this guy. And so that's going to cause that rolling, that tumbling motion. And what is that called?
Margination.
Yes, margination, page 343.
What is that?
It's the wheels turning.
So what it does, it's gonna start, it wants to start pulling these guys down, put them in the right area so that we can start moving these guys down and out. But it slows their response through here so that they will slow down to come into the fight. Okay, because we got to bring them into the fight. So basically what the book says, it changes the direction. Instead of having these guys flow through, it says that they usually travel towards the center of the vessel and settle towards the sides as the blood flow slows. So it's changing their direction. All right, so that's margination. Now the process of pulling these guys down and get them out here where the bacteria is. What is that? Yep. And is it. Very good. Okay. Once they're out here, we have our bacteria here. Our white blood cell here. How does the white blood cell know how to find the bacteria? It does follow the mediators. And what is that definition called? What's that? Chemotaxis. Our chemotaxis is going to lead this guy to that white blood cell. Now, in order to get some help, we need some help here. What is that terminology called? Very good. Interleukin 1 that actually calls and brings help from other white blood cells. Pretty good?
Oh, yeah.
All right, we're going to lunch. I'm not deleting anything, and it's recorded on YouTube. Let's see here. Hey Dixon, can you come over a second? So we're going to lunch. Come back at 13. Y'all need it. And I need to get my damn phone stopped beeping. I mean, they're blowing it up today. And it's people I can't ignore. Like, there, great. No, I can't tell them to stop, but I wish I could, or I would. Dix, that's exactly why I'm bringing Dixon up here. He's a strong arm.
I got your watch. I can tell. You can tell the phone is not being.
Oh, what commercial is that? The strong arm?
John
Boy.
Strong.
I'll let you go.
I did. He looked straight at me laughing, but I got lost in whoever said the super. I who did that. He looked straight at me and I was laughing. And he thought I was laughing at him, but I was laughing because of that. And you said there was like a backup. I was so moved. And I was out, man. He was like, no.
Thanks so much.
You got me good on that.
I just want to use it.
All right, so Barker, if you want to dismiss everybody, 1300, and that gives y'all plenty of time. I'm gonna let everyone 15 minutes early, but it'll let you. Well, let me ask this: is there any questions you guys have that you want me to fill before y'all go to lunch?
What's the real name for flags?
Okay.
Protein or something.
No, it's got a different name, but I don't think it's receptor. It's called something.
Protein.
The marker receptors, they love protein something.
I'll remember that.
And I'll get it for you when you come back for sure.
Like if you run into him, you're gonna.
This is all regarding inflammation response.
Yes. All this is the inflammatory response.
It's not you got like a decent diagram right on page 4.
Yeah, the book's pretty good. The book's good. I mean, for the most part, the book's good. It's just so like besides margination and transmigration, that's hard to get out of that book. You know, you wouldn't know really just from reading it. You need somebody to show that to you, but it's pretty good.
You said this
So this could be the Anaphylaxis response if you multiply it by about 100, right? Because what happens is that basophil comes along and sees this invader it's been looking for him since he left the first time. And now he just overreacts to it. And so you get a lot of vasodilation to the point where most of your plasma is in your feet. Now you're getting those leukotrienes we're going to talk about. They're the ones that cause bronchoconstriction and airway edema. Right now, leukotrienes like this, they're not going to cause that in this case. They might cause some pneumonia, and might cause some bronchoconstriction and stuff, but not like they do in an anaphylaxis case.
So this is if you get a cut, you get some dirt in it.
You get a cut, you get a strep throat, you catch pneumonia, you have any type of insult to the cell. So no, what they do is that they don't change their DNA, but they take that bacteria, change it to a antigen, and make it, and they put it up on one of those protein receptors, and just leave it there. Absolutely. Or if you get immunizations, that's the same thing for that too. That immunization works. You just don't get enough of it to cause you to catch the full disease. There's a little bit of
Go to 1040. I'm sorry.
No, don't do it.
no, come on, man. That's what I'm doing.
Don't do it. Why is it all the way?
I knew that was the one. Those are 4 different chemical mediators. Yeah. Leukotrienes and histamine are kind of like brothers and sisters, so they cause some of the same stuff.
He did it. I got reading it. Said, I got confused. I think it's poetry. But when I saw it, that's they all have
Their own thing.
Doesn't matter. Very, very, very minute details. Doesn't matter.
They'll have their own different receptors.
That's what they said.
Transmigrations are.
I understand.
Come on,
So this is something like they're not going to ask you on registry what a leukotriene is, right? But what this will do is it'll help you better see when you have somebody having an anaphylactic reaction or somebody who has pneumonia, who's sick. We'll explain how fevers develop from this. It'll help you to understand what's happening, right? So it's not necessarily they're not gonna like get you on registry with this, but it'll help you to have a better, broader look at some of the disease processes we're dealing with. Yeah, it's necessary. And something else too, it's necessary is because every program sets you up to go to the next step. They're providing information for you, just like we did EMT to come to AEMT. Like we talked about chemoreceptors, and we talked about aerobic and anaerobic metabolism, right? But then in A, we put acid base on it, right? We didn't cover acid base like that in E, but A we did, right? So in paramedic school, they'll take this a little bit further. There'll be another piece of this added. And so that's why it's important that I have to present it to you, because one day you might go to paramedic school. If you do, I need to prepare you for it. Otherwise, I wouldn't cover it. If I was just wanting you to pass the exam, I would just skip every bit of this. Because I don't think you necessarily need it to pass registry.
You're gonna email us, start up paramedic class. We want you.
Y'all want me?
We want you.
See, but you don't know any other instructors. I might compare to some other ones that suck. So,
We don't want any of this.
well, I just got, I don't know. My phone's been going crazy because we were awarded that grant.
What's that?
For the paramedic grant,
Oh,
we won it.
Oh, you gonna pick some-
nice.
So you guys are in this.
Not good.
I don't think, I still don't know. I don't know. I don't know. Stuff I have to do a lot different. A lot different. So I don't, I'm still, even though it's nice to have worked on a project and you, yeah, if we were looking at
Consider that.
as far as actually putting the leather to the road, I don't think we're close at all. I don't even think we're close at all. Yeah, exactly.
Everyone's gonna drop out. We gotta go to paramedic. No.
I don't, it won't be, if we do anything, it would be, it would, yeah, probably people from the field that go to paramedic school and do it in their off time. I would think. But this place is always off the chain. As soon as you think you've got something figured out, it's, they'll be telling me, hey, keep class 117, they're going to paramedic school. I mean, I don't think that would happen, but I'm saying sometimes off the wall, like right when you think you've got a plan figured out and you kind of know what they're gonna do, the exact opposite. I mean, not just opposite, it's a thought that never even occurred to you.
You can do fire first. I'll stay for you. I gotta get to fire. The beautiful thing is, you got the government on your side. So the grant just got approved. So even if
This
they
is
want to do
how
something
it
with it,
works positions.
even on their, even
But
if.
no, for real, I mean,
their highest
there's a
productivity
lot,
level,
there's a
we'll be
lot
ready
that
to
goes
just
on.
come next year.
It's crazy because who would have thought? Oh, to work at the fire department, you would need damn near an associate's degree just in EMS before you can even go train for fire. Who would have thought? Man, really? Well, hell, that's what you're getting right now. I mean, basically, I learned all this in college. And wasn't forced to learn 90% of the other stuff that I forced down your throat. I just had to learn this in one class.
This whole thing, a whole semester, one subject.
Yeah.
College for like paramedics, we're 2 years in. We have 2 years of training.
Yeah, some places absolutely. Some places you get.
Oh, yeah.
Yeah, there's a bachelor's in EMS and paramedicine right now. There's an associate's in paramedicine. So it would be great to get, if there's a way that we can hook up with another college, they might accept this as college.
That's crazy as hell.
Hey, Quinn. Mace.
Yes, sir.
Don't want to go to lunch? Come back at 13.
Okay.
Sound good? Maze, you awake?
Yeah. Stop that share.
All right. I think I'm gonna close YouTube down.
Yeah, we'll close you.
Probably a good idea. Yep.
Just saying. Still live.
I'm gonna change it.